Hi, today our group will be doing a small video presentation on spinal reflexes and reaction times in human. So now let's look into the objectives first. The objective of this experiment is to investigate the reflexes and reaction times in response to a variety of stimuli and under a variety of conditions. The second one is to determine some simple complex reflexes from an observer and measure the reaction time from an observer given harmless visual and sound cues. The third one is to study the time required for planned voluntary response to a cue. So now let's look into the introduction. Spinal reflex control allows our body to react automatically without the effort of thought. The reflex arc is a nerve pathway involved in a reflex action. In vertebrae, most sensory neurons do not pass straight to the brain, but synapse in the spinal cord. The reflex arc involves two neurons, which are sensory neuron, an intermediate neuron, or known also as interneuron, and a motor neuron. The reflex begins with a stimulus such as cold wind or heat from a stove, followed by a receptor. A receptor is how we respond to the sensory information and transmit the signal to a sensory nerve. Then a sensory neuron, which passes the nerve impulse to the spinal cord. The next is an intermediate neuron. This links the sensory neuron to the motor neuron in our spinal cord. A motor neuron carries the nerve impulse from the spinal cord to a muscle, and finally an effector, the muscle that is stimulated to produce an effect. Reflexes allow our bodies to protect themselves with fast reaction times using quick automatic processing. Reaction time is the amount of time required for the nervous system to receive and integrate incoming sensory information and then cause the body to respond. The simplest example of reaction time is the time required for a simple reflex to occur. The presence and strength of spinal reflex control is a significant sign of a nervous system development and function.
From experiment 6.2, we can conclude that the time lapse for each trials differ from each other, and the trials that recorded the lowest time lapse is the trials 8, which is 0.04 seconds, and the trials that recorded the highest time lapse is trial 7, which is 0.4 seconds, and the average reaction time of subject to the site is 0.19 seconds.
First, both experimenter and subject should have a stopwatch each and start both watches simultaneously. The subject should close her eyes. Next, the experimenter presses her watch to stop and simultaneously stepping the subject's shoulder. Then, the subject then presses her stopwatch to stop. From experiment 6.3, we can conclude that the time lapse for each of the trial varies. The lowest time lapse in the experiment is on trial 4, which is 0.06 second, and the highest is on trial 2 and 9, which is 0.16 second. The average reaction time of subject to touch is 0.12 second. Next, from table 6.3, the length of nerve pathway from shoulder to finger is 1.15 meter and the speed of the impulse transmission is 9.58 meter per second. In this experiment, students were required to swallow saliva in their mouth cavity to investigate the swallowing reflex whether it is completely voluntary action or not. From result and observation, it can be seen that as the student tried to immediately swallow the saliva again, he found that it is difficult to swallow for the second time. This indicates that saliva acts as a stimuli in our mouth cavity. When swallowing occurs for the second time, there is no more saliva, which means there are no more stimuli to stimulate the muscle in the throat. As for drinking water, it is found that swallowing the water became easier as the water replaced the saliva in our mouth. Precaution that we need to take in this experiment is students were advised not to put too much force in swallowing process as it can cause injury in the throat. Next, in experiment 6.2, we measured the time lapse between the time taken for experimenter to stop her stopwatch and the subject's time to react to the experimental action by stopping her own stopwatch too. From the observation and results, we can conclude that the reaction time varies for each trial and that the subject became more wary of the experimental action. Thus, there were certain trials with the lowest time lapse recorded, which is in trial 8 and trial 9. This indicates that the experimental action is the stimulus for the subject to take action. As the subject eyes detect the stimulus, the receptor on her eyes send the impulses to the brain that later were sent to the effector, which is the thumb to stop the stopwatch. Precaution that we take in this experiment is both the experimenter and the subject should have stopwatch in this experiment to avoid error in recording the time lapses. Lastly, in experiment 6.3, we measured the time lapse between the time taken for our experimenter to stop her stopwatch and the subject's time taken to react to the touch. The experimenter tapped the subject's shoulder and the subject will react to it by stopping her stopwatch without looking at the experimenter. From the observation and results, we can conclude that the time reaction varies for each trial. Without looking at the experimental action directly, the subject needs to rely on her receptor on her arm only. The tapping or touch sense on the subject's shoulder act as the stimuli that then were detected by the receptor. The receptor then transmits the impulses to the brain and the brain processes it and later transmits the impulse to the effector, which is the thumb, to react by stopping the stopwatch. The speed of the reaction depends on the cerebral reflex of the brain to process the information. Precaution that we take in this experiment is both the experimenter and subject should have stopwatch in this experiment to avoid error in recording the time lapses.
In conclusion, a spinal reflex is a reflexive action mediated by cells in the spinal cord, bypassing the brain altogether. All of the three experiments that have been conducted do not just simply to test only a simple reflex. Instead, it is to measure how fast our body reacts to the stimuli and how to measure the response to the time for something you can see or feel. Hence, the speed of our reaction plays a big role and an important part in our daily life. The reaction time is a measure of the quickness with an organism responds to some sort of stimulus.
That's all from Group 5 for Lab 6 Spinal Reflexes and Reaction in Humans. Thank you for watching.